Welcome to SWAYAM NPTEL course on Food Microbiology for Safe and Sustainable Food Systems. In the last class we have been discussing food-borne diseases and food safety in India. Now we will be discussing mechanism of microbial control in food, and in that we will be discussing thermal processing of food, and in this we will be discussing blanching, pasteurization and appertization. And the topic which we will be covering in this lecture would be introduction, thermal preservation of foods, methods of thermal treatment, blanching, pasteurization and appertization. In many cases, the microscopic flora creates serious problems in our food. The problems associated may fit into two categories: food spoilage, which occurs when the food becomes unpalatable due to microbial growth and develop undesirable flavors, odors, appearances or textures; and food poisoning, which occurs when the organisms present in food cause human illness or death through a toxin resulted into intestinal infection. Therefore, to avoid both of these problems, we need to understand the techniques which prevent their growth. Thermal preservation of foods. The most common method of killing microorganisms is to subject them to a heat treatment. High temperatures act by killing vegetative cells and also spores and denaturing the food enzymes. It may also act to destroy toxins produced by certain microorganisms. The heat treatment used depends on the following factors: what time temperature combination is required to inactivate the most heat resistant pathogens and spoilage microorganisms. The higher the temperature, the higher the temperature, the less time needed, and vice versa. What are the heat penetration characteristics of one particular food and container of choice if it is packaged? What are the types of microorganisms present in the food material? The thermal death time of different microorganisms vary widely with the species. What is the concentration of the microorganisms? The higher the concentration, the more time is needed. What is the state of the microorganism? Spores are more resistant than vegetative cells. What is the effect of various environmental factors such as food acidity or pH and salt or solutes? There are two main temperature categories employed in thermal processing: pasteurization and sterilization. The basic purpose for the thermal processing of food is to reduce or destroy microbial activity, to reduce or destroy enzyme activity, and to produce physical or chemical changes to make the food meet a certain quality standard. Methods of thermal treatment. Mild processes, that is blanching and pasteurization. Heating a liquid, particularly milk, to a temperature between 55 and 70 degrees Celsius to destroy harmful bacteria. More severe processes like canning, baking, roasting and frying. And sterilization, that is complete destruction of microorganisms, heat resistant spores by treatment of at least 121 degrees Celsius of wet heat for 15 minutes or its equivalent. Now, blanching. The primary purpose of blanching is to destroy enzyme activity in fruit and vegetables. It is not intended as a sole method of preservation, but as a pre-treatment prior to freezing, drying and canning. Other functions of blanching include reducing surface microbial contamination, softening vegetable tissues to facilitate filling into containers, removing air intercellular spaces prior to canning. Enzymes are proteins which are denatured at high temperatures and lose their activity. Enzymes which cause loss of quality include lipoxygenase, polyphenol oxidase, polygalacturonase and chlorophyllase. Heat resistant enzymes include catalase and peroxidase. This is the pictorial representation of kitchen terms. That what to know about the blanching. In the first step, the foods are plunged into the boiling water and then in ice water. What it does? It sets the color of vegetables, allows for easy fruit peeling and loosens skin of nuts, and food does not cook all the way, so texture is preserved. It serves as the first step of food preservation by denaturing enzymes. Now, methods of blanching. Blanching is carried out at up to 100 degrees Celsius using hot water or steam at or near atmospheric pressure. Fluidized bed blanching, utilizing a mixture of air and steam, has been reported. Microwaves blanching, this includes rapid heating and less loss of water soluble compounds, The steam blanching, a preferred method for foods with large cut surface areas as lower leaching losses. Individual quick blanching involves in single layer of the food is heated to sufficient temperature to inactivate enzymes, and a second stage in which a deep bed of the product is held for sufficient time to allow the temperature at the center of each piece. Hot water blanchers includes various designs which hold the food in hot water at 70 to 100 degrees Celsius for a specified time, then moves it to a dewatering or cooling section. Testing of the effectiveness of blanching. Over blanching causes quality loss due to overheating, while under blanching causes quality loss due to increased enzyme activity, because enzymes activated and substrates released by heat. The peroxidase test in vegetables is used to detect enzyme inactivation. This enzyme is not in itself implicated in degradation, but is relatively heat resistant and easily detected. It consists of adding guaiacol solution and hydrogen peroxide solution and observing the development of a brown color indicating peroxidase activity. Complete inactivation is not always essential. Green beans, peas and carrots with some residual peroxidase activity have shown adequate storage quality at -20 degrees Celsius, though with other vegetables, for example Brussels sprout, zero peroxidase activity is essential. Advantages and disadvantages of blanching. Advantages include faster, more uniform heating, good mixing of the product, reduction in effluent, shorter processing time, and hence reduced loss of soluble and heat sensitive components. Disadvantages include high capital costs and potential difficulties in uniformity of heating. Now, pasteurization. Pasteurization is a relatively mild heat treatment in which food is heated to less than 100 degrees Celsius. It is widely used throughout the food industry and is frequently employed as a critical control point in various HACCP plants. As a unit operation in food processing, it can be used to destroy enzymes and relatively heat sensitive microorganisms, for example non-spore forming bacteria, yeast and molds. In this regard, it is used to extend shelf life by several days, for example milk, or months, for example bottled fruit. The severity of treatment and resulting extension of shelf life is determined mostly by pH of the food. If low pH food, that is pH less than 4.5, the main purpose is destruction of pathogenic bacteria, while below 4.5 the destruction of spoilage microorganisms or enzyme deactivation is usually more important. The extent of heat treatment required is determined by the D value, that is decimal reduction time or time to reduce numbers by a factor of 10 or 90% of the initial load of most heat resistant enzyme or microorganism which may be present. In terms of checking the effectiveness of the process, alkaline phosphatase is a naturally occurring enzyme in raw milk with a similar D value to heat resistant pathogens, and so is routinely used as an indicator of adequate pasteurization. If phosphatase activity is found, it is assumed that pasteurization is inadequate. Pasteurization is normally used for the destruction of all disease causing organisms. For example, pasteurization of milk, or the destruction or reduction in the number of spoilage organisms in certain foods, for example vinegar. This table shows the pasteurization temperature and the time required. In the wet or batch type of pasteurization, the temperature is 63 degrees Celsius and the time required is 30 minutes. But in the HT/ST pasteurization, that is high temperature short time pasteurization, if the temperature is kept 72 degrees Celsius, then time required is 15 seconds. If the temperature is increased to 89 degrees Celsius, then the time required is 1 second. If the temperature is increased to 90 degrees Celsius, then it is 0.5 seconds. If it is further increased to 94 degrees Celsius, then time required is 0.1 second. If it is increased to 96 degrees Celsius, then it is 0.5 seconds. Then it is 0.05 seconds. And if the temperature is 100 degrees Celsius, then the time required is 0.01 second. These temperatures are equivalent and are sufficient to destroy the most heat sensitive of the non-spore forming pathogenic organisms. Milk pasteurization temperatures are also sufficient to destroy all yeasts, molds, gram negative bacteria and many gram positive. The two groups of microorganisms that survive pasteurization temperatures used in milk are thermoduric. Thermoduric are organisms that can survive exposure to relatively high temperatures, but do not necessarily grow at these temperatures. For example, Streptococcus and Lactobacillus. The another group which survive pasteurization is thermophilic. These are organisms that are not only survive relatively high temperatures, but require high temperatures for their growth. Now, methods of pasteurizing. There are number of basic methods of pasteurization widely used in the industry. The first is batch or holding type. In this method, every particle, for example milk, must be heated to at least 63 degrees Celsius and held for at least 30 minutes. However, this is not used commercially these days. Now, low temperature long time process or LT/LT process, where pasteurization time is in the order of minutes and related to the temperature used. Two typical temperature time combinations are 63 to 65 degrees Celsius for 30 minutes or 75 degrees Celsius over 8 to 10 minutes. Now, pasteurization temperature and time will vary according to nature of product, initial degree of contamination, and pasteurized product storage conditions and shelf life required. In LT/LT pasteurization, it is possible to define three phases: heating to a fixed temperature, maintaining this temperature over the established time period, that is equal to pasteurization time, and cooling the pasteurized products by natural, that is slow process, or forced cooling. This is a typical batch method where a quantity of milk is placed in an open vat and heated to 63 degrees Celsius and held at that temperature for 30 minutes. Sometimes filled and sealed bottles of milk are heat treated in shallow vats by that method and subsequently cooled by running water. Now, high temperature short time or HTST process. High HTST pasteurization is characterized by a pasteurization time in the order of seconds and temperature of about 85 to 90 degrees Celsius or more, depending on holding time. Typical temperature time combinations are as follows: at 88 degrees Celsius for 1 minute, at 100 degrees Celsius for 12 seconds, and at 121 degrees Celsius for 2 seconds. Now, ultra high temperature or UHT pasteurization. The UHT involves exposure of a brief, intense heating, normally to temperatures in the range of 135 to 40 degrees Celsius, but for a very short time, a second or even less. The process depends upon a fairly complicated sterilizer or aseptic filling design. The two stages of effective heat sterilization followed by aseptic filling represent an integral system. Frequently, the packaging material for UHT milk is cardboard, which must be chemically sterilized prior to the filling operation. This is the pictorial representation of pasteurization process. When the milk is flowing, then enzymes are in full working conditions. When it passes through the heating chamber, the enzymes go through heat treatment and they are denatured and becomes unable to function properly. And after heating section, when it comes to the cooling sections, the enzyme continues to the cooling treatment and is still unable to function in the process of helping bacteria to grow. These are the differences among the different pasteurization methods. Like wet pasteurization, the milk is held at 63 degrees Celsius, milk is heated to 63 degrees Celsius for 30 minutes, whereas for the eggnogs it is 69 degrees Celsius for 30 minutes. The eggnog is a rich chilled sweetened dairy based beverage traditionally made with milk, cream, sugar, egg yolk and whipped egg white. In the high temperature short time, that is the HTST process, milk is heated to 72 degrees Celsius for 15 seconds, whereas for eggnog at 80 degrees Celsius it requires 25 seconds of heating, whereas 83 degrees Celsius requires only 15 seconds. In another process of pasteurization, that is higher heat shorter time, that is HHST, the temperature is further increased to 89 degrees Celsius, then only 1 second is required to heat. At 90 degrees Celsius 0.5 seconds, 94 degrees Celsius 0.1 second, 96 degrees Celsius 0.05 second and at 100 degrees Celsius 0.01 second. Whereas in ultra pasteurization, which is generally used for milk and cream, The temperature used is 138 degrees Celsius for just 2 seconds. Now the appertization. It refers to processes where the only organisms that survive processing are not pathogenic and incapable of developing within the products under normal conditions of storage. As a result, appertized products have a long shelf life even when stored at ambient temperatures. The term was carried as an alternative to the still widely used term commercially sterile, which was objected on the grounds that sterility is not a relative concept. A material is either sterile or not sterile. An appertized or commercially sterile food is not necessarily sterile, that is, completely free from viable organisms. It is, however, free from organisms capable of growing in the product under normal storage conditions. Thermophils may be present but will not grow in the ambient temperature. This is the appertization process. Like, what is appertization? The appertization is a food preservation method developed by Nicholas Appert in the 19th century. It involves sealing food in airtight containers and heating them to destroy bacteria, enzymes and other microorganisms, ensuring extended shelf life without refrigeration. Now, how it works? It requires sealing, that is, the food is placed in airtight containers preventing the entry of air and microorganisms. Then comes heating. The sealed containers undergo heat treatment, typically through boiling or steam, to eliminate harmful bacteria. The next step is cooling. After heating, the containers are rapidly cooled to prevent recontamination. And sealed and sterilized foods can be stored for an extended period without spoilage. And why appertization? That appertization revolutionizing the preservation and preserving nutrients.